First, I would like to say some words on engineered nanomaterials as analytes. It means subject of chemical analysis. Then I want to mention the parameters prescribed by Uniform Description System for nanomaterials on nanoscale. And finally, analytic chemistry of nanomaterials and bioanalytical chemistry of nanomaterials, particularly with respect to the assessment of toxicity risk. I will finish with some ideas regarding the outlook. As we probably know, nanoscale materials are distinguished as materials with at least one dimension between 1 and 100 nanometers. They are generally divided into nano-object and nanostructure material. Regarding nano-object, according to the nano dimensions, we speak about nanoplates, nanofiber, nanoparticles. With 2 dimensions, there are nanotubes, as we have heard in previous lecture. Regarding the composition and form, nanoparticles can represent either single material or composite material or various more complex materials composed of core and shell or functional groups on the surface. I have mentioned already analytes. It means species which are subject for chemical analysis. And here on this slide, I want to highlight the differences between molecular and nano level. Typically, we speak about determination of single molecules, analytes represented by single molecules. In the case of nanomaterials, structure I have mentioned, we have assemblies of 100 to 100,000 molecules, while it's single molecule at molecular level, the structure, it means geometry, is important parameter. At nanoscale, we speak about morphology of nanoparticles and engineered nanomaterials. In difference to molecular level, which define stoichiometry, in the case of nano level, we have typically not defined stoichiometry. And together with chemical properties, surface properties become of really very, very important. That is, that not only intramolecular, but also intermolecular interaction play a significant role. Also, such properties as electric conductivity and some other connecting with electron delocalization within the single molecule, in the case of nanomaterials and nanoparticles, the ohmic properties represent the dominant background for electric conductivity and other physical properties. In 2015 and 2016, Uniform Description System for materials on the nanoscale have been prepared by the CODATA organization. It once more definition which is the same or accepted by ISO and European community. And this system describes not only parameters how to characterize nanomaterials, but also concerns production specification and general identifiers of nanomaterials. From the point of view of distinguished nanomaterials, They are considered nano-object as individual particles, collection of nano-object, and then bulk materials. Bulk materials with identifiable nano-objects and with bulk materials with features in the nanoscale. Regarding chemical analysis, in both cases, nano-objects and collection of nano-objects, the chemical composition is considered as a parameter necessary to be considered for characterization of materials, together with crystallographic structure and morphologic parameters, and with interface and intermolecular interparticles interactions. The system named a rather large scale of physical and chemical methods for characterization of these materials. From physical, it is size, shape, association, agglomeration, state, crystal structure, and surface chemistry. I'm chemist, so I'm speaking about analytical chemistry. What we are able, it is to characterize, determine solubility and stability of nanomaterials in various media over time, and of course, elemental composition, changes in elemental compositions. This method can be separated according to parameters of the characterization, and I like this picture when we can see on the left hand side method connecting with concentration, surface, and composition properties, and on the right hand side there are methods characterizing more or less physical parameters of nanomaterials. The progress within the last decade can be demonstrated by a large number of books and excellent review articles. Some of them are mentioned here. Each of them deals with the characterization of nanomaterials both from the physical and chemical point of view. And the development on this topic continues, comparing the present and future concepts with the history of chemical characterization and particularly characterization of nanomaterials. One of these reviews named size and concentration ranges reachable by individual techniques. In 2008, Hasselhoff has formulated task for analytical chemistry for this decade. There are not only the question of chemical analysis, but also steps of the analytical process which significantly can influence the result of chemical analysis. First of all, it is generally in analytical chemistry sampling and sample handling. So how to obtain nanomaterial from such complex matrices like environment, water, soil, air, from consumer products, particularly from cosmetics, from food, and finally from living organism. After extraction and separation of this method, also fractionation using various physical approaches takes important. Nanometrology, it means metrological parameters developed for the analysis at the nanoscale, is also undergoing huge progress and will do significant step, as it seems, on the development and proposal of new reference material and reference methods. Validation and determination of measurement uncertainty together with the results is typical for molecular level of analytic chemistry, but should be developed also for nanoscale. I have mentioned complex matrices, species analysis, enviro and bio distribution of nanomaterials that are topics which become really important, particularly from the point of view of end users and human community. I have the possibility to be a chairperson at the current IUPAC, International Union for Crude and Applied Chemistry, large interdivisional project, Analytic Chemistry of Nanomaterials, Critical Evaluation, which should be published within 1 year, and when we deal with the present state of the art, particularly on this new task. Together with the preparation and application of nanomaterials, the people consider the risk, potential risk, particularly with respect to their toxicity, and methods for assessment of such toxicity risk are subject of scientific conferences, papers, and even journals. It is due to possibility of various nanomaterials, carbon-based, silicon-based, metal-based, to enter the living cell and to damage biomolecules and biopolymers, particularly due to production of reactive oxygen species. In this respect, all three aspects of nanomaterials are important and play significant role, not only chemical composition, but also size and shape properties, it means physical properties of nanoparticles and engineered nanomaterials, and their surface modification, surface properties. With respect to importance of such studies and approaches to particularly human health, there are large international activities on this topic. Here I mention some projects or name of activities by Environmental Protection Agency, by OECD in Europe, by European Union regulations, and finally also by the Union of Pure and Applied Chemistry, where 2 years ago really large technical reports have been published regarding the preparation and characterization, I mean physical characterization of nanomaterials, and also nanotoxicology and application of nanomaterials with respect to human health. I have mentioned the publication activity on this topic. Here you can see again a selection of largest reviews or book publications regarding the progress in the evaluation of potential risk properties. Cytotoxicity is typical methods considering the destructive action of nanomaterials on certain cells. For the series of similar nanomaterials like nanotubes, quantum dots, etc., computational methods have been developed, for instance, by Dr. Puzin or Richards, which are able to predict risk properties within the series of nanomaterials. Bioanalytical methods, where I am active, try to evaluate damage to some biopolymers, biomolecules, mostly to deoxyribonucleic acid, using various bioanalytical approaches, comet assay, geoelectrophoresis, but also combined methods, gas chromatography, mass spectrometry, liquid chromatography, mass spectrometry. Not only in vitro, but also in vivo toxicity studies are started today and are needed for complex evaluation of side effects, unwanted effects of nanomaterials. Not only nucleic acid, but also proteins within proteomic studies and beta-molomic studies start to be applied. When speak about my laboratory, we develop biosensors, nucleic acid-based biosensors, that on the surface of transducer, the biorecognition element, in this case nucleic acid, is attached together with various nanoparticles, quantum dots, metallic nanoparticles. And we use two approaches, typical biosensors, detection rates and biosensing. It means in testing of interaction between biomolecules and nanoparticles in solution, and then using biosensor or chemical sensor for evaluation of these properties. Here I would like to demonstrate only one of the results obtained with quantum dots based on cadmium sulfate or cadmium tellurate, when we can compare and see the effect of the fluorescent quantum dots present in such study, comparing the red and the green columns, detecting the survived portion of DNA. We can see that in the case of highly fluorescent quantum dots, the portion of survived DNA under UV irradiation significantly decreases, and using other detection approach, it means signal responses of DNA bases, we can see not only decrease, but partially or initial increase in the responses and then decrease. It means that these approaches can, on the molecular level, totally other than detection using cells, indicate the processes and changes in the structure of biomolecules. Going to summary, I would like to stress that nanomaterials become more and more important subject of chemical analysis. Once more, for their characterization as products of nanotechnology, and also for the detection and evaluation of the risk for environment, for food, for consumer products and human health. Not only characterization, but nanomaterials are used in analytical procedures as important components of analytical devices and procedures, both as interfaces of detection equipments and as labels producing signals. In development of analytical chemistry as a science, we see also a strong influence of nanotechnology and nanomaterials. New branches of analytical chemistry appear within last years: analytical nanoscience and analytical nanotechnology, nanoanalytics. I mentioned already analytical nanometrology, production and use of special reference material and validation procedures. Nanomaterials become subject for evaluation of toxic properties, potential toxic properties, as I stressed, as I have mentioned. To new fields, we can mention also nanoethics, which compare the benefit and risk of nanotechnology. All these new discipline and new branches of development probably will reshape understanding of nanoparticles and nanomaterials and influence our legislation. Thank you very much for your attention.